everyone, welcome to the Quantum Bridge. And today we have a guest from New Zealand, I believe, and his name is Keryn. Welcome to the show, Keryn.
Thanks, Gary.
Now, Casper, what are we talking about today? Something sweet, I believe.
Let's see where we go, because Dr. Keryn Johnson is a man who has many different facets, and is incredibly steeped in really a new vision, I think, for me at least, of the way science, I hope, would be moving and unearthing rather special things. But we're going to try to keep this particular recording to talking about a specific honey that Keryn is involved in. And I'm going to say hello, Keryn, and I'm going to pass across to you to do any sort of intro on yourself, or immediately leaping into the honey area that you'd like to do.
A little bit of background from me. I have 4 degrees, 18 years as a research scientist in a New Zealand government organisation, and I've worked with honey really since 2010, and in particular Manuka honey. So I think today we're going to try to curtail my creativity and focus in on really the functional benefits of Manuka honey. Okay, so I can start with a little story that goes along the lines of Manuka honey special. Manuka honey special because it's got this compound called MgO, or methyl glyoxal. And the honeybees, westernized honeybees that have been introduced to New Zealand, collect the nectar from the Manuka plant. And in doing so, they've kind of carried a compound called dihydroxyacetone, DHA. And the DHA is modified during maturation or during storage of Manuka honey to produce this compound called methyl glyoxal. And methyl glyoxal has been shown to have antibacterial properties, and it's those kind of antibacterial properties that make Manuka honey special. So that's the story that I guess the New Zealand industry has been selling to the world.
It's native to New Zealand.
And as a scientist,
it doesn't come from other countries, does it?
Well, there are Manuka plants over in Australia.
Okay.
It's not limited to just New Zealand, but it's considered a tonga in New Zealand. It's a precious gift, right, from this land. The word Manuka is Maori, and so we've kind of got this kind of indigenous and Western kind of coming together, right, in New Zealand. And that's really the Western honeybee also being introduced to New Zealand and collecting pollen from indigenous plants. So the story that the industry has been telling and marketing is not quite right, and that's it's a difficult pill to swallow when the marketing is done in a way to try to simplify things for the general public so that they can understand. But science also advances and moves on, and sometimes when new discoveries are made, the old story has to be changed. And so there's this kind of problem of, well, we've been marketing, how many millions of dollars is New Zealand making from Manuka honey? And there's a whole heap of other problems about adulteration of Manuka honey overseas. And if you have a look in some of the, I guess, the scientific journals, you would see that, well, really, we don't know what the unique Manuka factor is. Peter Moland, who's really the godfather of I guess the Manuka honey story in New Zealand and its antibacterial properties suggests there's a synergistic benefit of other compounds plus methyl glyoxal. Okay, so that's kind of the background setting of the Manuka honey story and the international marketing of Manuka honey globally. And then I kind of come into the picture in 2010 and start looking at Manuka honey for a company. And that company just really wanted me to repeat what Peter Molin's team had done at Waikato University. Guy Henley out of the UK discovered MGO, so that wasn't discovered in New Zealand at all. And there's always been this dispute going on, and that's typical of science, right? Science is not something that's concrete.
Yeah.
And when you do things from different perspectives, you often see different outcomes. And so this whole kind of dihydroxyacetone being converted into methyl glyoxal as part of the, I guess, the maturation of Manuka honey, and the older it gets, like a good wine, it gets better and it gets a higher concentration of methyl glyoxal, so it's worth more. So the industry stores honey for a long period of time, can be up to a couple of years, to increase the methyl glyoxal content in order to get a higher value for the product.
Is this what we see with the plus 10
think,
or plus 15 and the sort of concentration of these products?
Yeah. So there's a, I guess, a number of criteria that the New Zealand government has requested the honey industry measure in order to make sure that it is, I guess, acceptable to the consumers. So they have a methyl glyoxal measurement, they have a dihydroxyacetone measurement, they can have a pollen DNA measurement, and they also have an HMF, which is another compound which accumulates if the honey's heated. So there's all these kind of criteria that is regulatory requirement. And so, honey is a very ancient product. It's been used for thousands and thousands of years. The methyl glyoxal, the MGO in Manuka honey, that destroys the activity of glucose oxidase, which is an enzyme that's present within honey. And so if there's no glucose oxidase, then the honey cannot produce hydrogen peroxide. And that's typically understood as the way that most honeys work to be antibacterial. So they destroy bacteria by hydrogen peroxide. But that's not quite right either, because the hydrogen peroxide reacts, and there's another kind of step in that pathway to produce something that's known as a hydroxy radical. And that hydroxy radical system has enough energy to break down bacteria back into CO2 and water. So it's not like a drug where you're inhibiting a particular enzyme that's responsible for forming the membrane or the cell wall of the bacteria. It's not working as that kind of mechanism at all. The way that honey works is it produces something that reacts with the bacteria, that destroys the bacteria and turns those structures back into CO2 and water. And that's really what this hydroxy radical system does. So because MGO inhibits this glucose oxidase to stop hydrogen peroxide being formed in Manuka honey, then the thinking was, well, it can't produce this hydroxy radical system. So what's the active ingredient in Manuka honey that makes it special? And that's what Henley found. He said, well, it correlates. It correlates with the constant methyl glyoxal, which has a correlation to antibacterial activity. It doesn't mean that MGO is the antibacterial compound. And if you look in the scientific literature about methyl glyoxal, it gets a little bit complicated because there's this whole kind of problem associated with methylglyoxal and diabetes and advanced glycation end products, which triggers inflammation. And so there's also been pushback in the marketplace saying that, well, methylglyoxal has these detrimental aspects to it. But you're saying Manuka honey is good because it contains high methylglyoxal. So science is never straightforward, right? The public's told a story through marketing. But when you see the complexities of what's actually going on, then you kind of start scratching your head and you go, well, if it's not methylglyoxal, then what is it in Manuka honey? And so I was, I guess, fortunate enough to kind of have the opportunity to explore that. And so what I did was I started from scratch, literally just kind of ignore what everybody else has done in the literature. And you start from a clean slate again. And you go, well, we know that hydroxy radicals are involved in hydrogen peroxide producing honeys through this high energy hydroxy radical system. And then, well, what are the processes? What are the components that are required for that? Well, hydrogen peroxide is one thing. That's not working in Manuka honey. So that's a problem. But that was more complicated because all of a sudden it's like, well, iron, it needs iron. Yeah, that's a tick. That's a high iron content in Manuka honey. It needs really the right pH for that kind of chemistry to occur, and Manuka honey's at the right pH for that. And so I started kind of exploring other kind of chemistries that could result in the formation of the hydroxy radical system. And what I discovered really was a process called photofenton chemistry, where light is involved. And so all of a sudden it was like, okay, so now we have iron and we have certain compounds, and there's a lot of phenolics, antioxidant type compounds in Manuka honey, as well as the methylglyoxal. So I started kind of playing around under a microscope to see which components of the honey could come together to produce these hydroxy radical systems using different colors of light. So blue light with its high energy, or UVA light with its high energy. And so all of a sudden I kind of stumbled upon the iron light that could trigger formation. And so the hydroxy radical system was being generated through kind of a holding hands of the MgO with an iron atom. And this light mediated system really gives processes operating very quickly at nanometers to produce these high energy radical systems that break bacteria back to CO2 and water, break down viruses, off inflammation. So it was like, okay, there's more to the story than we were initially kind of led to believe. And if you explore cell biology a little bit, you'll see that, well, methylglyoxal gets broken down by this enzyme system called the glyoxylase system in biology. And all cells have this kind of glyoxylase biochemical pathway that turns methylglyoxal into lactate, delactate. And all of a sudden it's like, okay, well, we're saying that Manuka honey has these benefits, but is it actually getting past the glyoxylase enzymology that's operating in our biology, or is it being converted into delactate? I don't think anybody has the answer for that. And so it's like, okay, orally, there's some benefits. What they are and how they are kind of imparted into the human after eating honey orally, really difficult to understand that complexity because there's obviously MGO. I was kind of really, I would say, somewhat confused, right? And you get a story and by the industry and they're saying, well, this is how it is. And they say, well, that glyoxal's healthy. And then this whole diabetes side of things is going, well, methyl glyoxal impacts on diabetes through this process where MGO reacts with amino acids on proteins that stimulates inflammation through certain receptors on macrophage, which is a white blood cell type. And then you have this other story of high energy radicals, the hydroxy radical system, which breaks things back down into CO2 and water, which is really a potent oxidant radical system, which if you read in the literature, you would say, well, that's really damaging to cells and it destroys cells. And so you kind of go, one story, MGO doesn't actually do much because it's getting broken down into D-lactate and it's taken orally and the biology is actually getting rid of that. And then there's this other story, well, it's putting a high energy radical into your body and aren't radicals bad? And so if you dig a little bit further, you can identify that, well, radicals are used by biology to do chemistry, not at the expense of using ATP. So it doesn't require ATP. The hydroxy radical has a half-life of a nanosecond. It's 1200 electron volts of energy. So the physics operating is very quick. So it can't migrate out of the cell. It's actually made locally and used at the site of production. And so when I was looking at, I guess, pollen grains in Manuka honey, I saw, I guess, a range of pollen grains that contained like a fluorescent ring inside them. And on one occasion, when I was doing some mass spec analysis, I discovered that inside pollen, there was a polymeric glyoxal material. And I found some literature that kind of told me that pollen germination requires hydroxy radicals in order to facilitate the growth of the pollen tube. So, I was led to believe that dihydroxyacetone collected from the nectar by the bees was responsible for methyl glyoxal formation. Now, that doesn't seem to be the case either. What appears to be the case is that this methyl glyoxal is coming from the pollen as part of pollen tube formation. So if you think about a pollen and everything inside it, you need to form water and have a system that pressurizes it to get the extension of the pollen tube growing out of that. And there's this light-based photophentin system that takes the phenolics in the pollen and adds hydroxy radicals to the ring and breaks it open and turns it into a polymeric material of glyoxal, breaks it down further into CO2 and water. So all of a sudden, well, it's not DHA coming from the nectar collected by the bees. It's actually the bees collecting the pollen that then undergoes a process of maturation and like pollen tube germination that's generating methyl glyoxal from that glyoxal polymer that's forming in there. And that's just like, well, oh, hold on. That's a different part of the story as well. So all of a sudden, we've got a high energy radical system that's operating in the pollen to form methyl glyoxal. And the industry know that high MGO Manuka honey has low pollen counts. They know that correlation, but they didn't put two and two together that methyl glyoxal was being generated from this process happening in the pollen grain. And so I've been, I guess, putting all this together, an alternative story of Manuka honey. And rather than stepping on the toes of the industry, which is selling honey, I've kind of gone off in a different direction. I've gone, well, I know in cell biology, phagocytosis uses process where it's producing hydrogen peroxide in a phagosome, and like this spherical vesicle. So when the bacteria gets engulfed by the macrophage, it exposes it to hydrogen peroxide, and some of the chemistry and physics operating in that little spherical vesicle is the formation of hydroxy radicals, which is breaking down the bacteria back into CO2 and water. So our bodies utilize hydroxy radicals to fight viruses and bacteria and fungi. It's what it does in order to really destroy pathogens. It doesn't try to stop them by putting in an inhibitor to an enzyme. No, it just kind of wipes the slate clean and completely kind of obliterates it. It's like it's a very unique system that's actually operating. And I found that hydroxy radical system is also operating in apoptosis, pre-programmed cell death. And so it's important for the turnover of our cells in our bodies to be able to break them back down into CO2 and water to excrete them, to breathe and to excrete them, right? So our bodies are doing that naturally. So all of a sudden it's like, oh, okay, if that's natural for human cells to utilize the hydroxy radical system, and that's working at nanosecond speeds, a billionth of a time per second, then all of a sudden I had to kind of consult biology, because it's used high energy physics to break stuff down, to recycle things. So that whole kind of death and regeneration system is operating using hydroxy radicals. And here's Manuka honey having health benefits that supports the history. So I put two and two together and really kind of figured out that, well, how do you stabilize something that's lasting a nanosecond, that uses light to stimulate the turning the atom of iron from an Fe3 to an Fe2. And when it's in that Fe2 state, it's called photoreduction. It's able to react with the hydrogen peroxide to produce the hydroxy radical. So it's a very kind of specific kind of process. So I was, I guess, lucky that I saw the literature and identified that, well, methyl glyoxal stops glucose oxidase, stops the production of hydrogen peroxide. So methyl glyoxal really stops that photophentin chemistry cycle at the point of photoreduction of iron, and there's no hydrogen peroxide for it to react with. So it's kind of stuck there in a photoreduced state. And so I figured out a way of really kind of purifying out the royal jelly protein. So it'd go part way along the process, but would stop because it wasn't exposed to hydrogen peroxide when I was doing the purification. So I was able to kind of recover the proteins, and that allowed me to kind of recover the antioxidants, which had the coordinated minerals attached. And it's that kind of coordination chemistry that's really responsible for the production of that photoreduction, the process of producing the form of iron coordinated to the antioxidants that makes it reactive toward hydrogen peroxide. And really, really fascinating is that these proteins are 10 nanometers in size. So they're really, really tiny. Get over 1 billion of them per milliliter, right? So we're talking about really, you would say, well, how much does that weigh? Well, if you freeze dried a mil, you might get 1 milligram. So 1,000th of a gram, but there's over 1 billion of these protein particles in that. So you go, oh, okay. So we're talking very, very small things. And the protein, because it's in honey and the bees have put it in there, and honey has a very low water content, it's actually hydrophobic, which means it doesn't like water. And it's quite weird. You dissolve honey up in water and you tip tip out the water and you'll see this glossy film around the inside edge of a glass or something. And you go, well, okay, what is that? Well, it is actually predominantly this protein is actually binding onto the surfaces. So I figured out how to really purify out the protein. And it was still, I guess I've done it in a gentle way. You know, I'm an enzymologist, protein biochemist. I figured out how to purify the protein gently. And in doing so, was able to recover the bioactivity that was associated with single atoms that were coordinated to the minerals, or the minerals and the antioxidant component, which was bound onto the protein. And the proteins were hydrophobic, so they were compatible with skin. So I started trying the product for myself as a topical product, rubbing it in as I purified it, just to see whether this was actually going to be beneficial or detrimental. See, you go, well, the literature says that hydroxy radicals are bad for you. And I'm going, well, hydroxy radicals are generated by Manuka honey, and Manuka honey's good. And then that whole apoptosis story suggests this death regeneration system. So I went, okay, I'm going to try it for myself, see what happens. And when I did, the first thing I noticed after about 25 minutes is I was buzzing. I was like very energized. When I just purified the product and used it topically, it was like, hold on a sec, what's going on? And so it was like, okay, like in a kind of slight yellowish color. Couldn't see any protein in there or anything. You do a UV vis absorbent spectral analysis, you can't see them. The wavelengths of light used to look at proteins around that UV 280 nanometers or 214 nanometers, because the particles are down at 10 nanometer size, they're too small. And you kind of, it's like working with the invisible. So I kind of had to figure out what technologies were suitable to be able to detect these proteins. And there's been a lot more literature kind of even since the 1930s. There was a couple of people over in the US who identified protein in honey back in the 1930s. And they used to remove it out of honey using bentonite in order to make the honey like sugar. So they removed all the proteins, which is kind of where all the activity is actually happening. But that's often the way. The consumers like a very nice, clear, white honey, and that doesn't darken on aging. And here's me going, well, actually, I'm just going to take the protein. That's the component that's actually functional because of the complexation that's occurred with the antioxidant and the mineral, and use it as a topical spray. So that's kind of really what I've been developing. And so when I do my purification, I'm utilizing that process where the pollen is germinating. I'm stimulating pollen germination. I'm getting MGO to be generated, which then reacts with the proteins to give the complexation, and that purify that out. So I've been able to kind of develop a method that gives me this bioactivity. And then I've been, okay, here are the tools. It's like, I need a nanosite microscope, which is like $300,000 worth of equipment, or a Zetasizer, or Izon in Christchurch here in New Zealand. They've got a Q Nano, essentially looking at things in the nanometer scale, tiny things. But I figured out that if I do a few things and I use the right pieces of equipment, I can actually monitor and figure out how much of these things I've got. But I remember making some batches and having 10 million particles per mil, and then this other product I was kind of playing came up and it had a trillion particles per mil. It was like, really hard to conceptualize how such small things can kind of be in such a volume, right? It's the mind doesn't really allow you to kind of figure that out very easily because it's not in a frame of reference that corresponds to a meter or a second. So all of a sudden you kind of have to go, what's the fastest thing in the universe, or what's the smallest thing in the universe? And most people will say, well, the speed of light's the fastest thing and the Planck length's the smallest thing. So when you go, oh, okay, fine, so at least you've kind of got some ideas relative to I guess the speed of light and how far that can go in 1 second. So when you're dealing
Keryn,
down
talking about the speed of light, we appear to have got through this at what feels like the speed of light. I just want to let everybody know that we will put the links to Keryn's wonderful products and the products that, as you will now see, have come out of this immense passion for the study around this and a much wider framework and frame of reference that Keryn operates out of, which we hope to get him back on to talk about for the audience at another point. But for now, Keryn, I mean, look, I know that this is tough to encapsulate. I think you've done a brilliant job, and we'd love to see you again. But in the meantime, I think that we have given, or you have given, a beautiful and very heartfelt overview of why you're so passionate and why this is something that you seek to put out into the world to help people. So I want to say thank you for that.
Thank you.
You're welcome. You're welcome.
We'll see you again.
So for now, from the Quantum Bridge, until next time, hopefully with Keryn, take care, everybody.
And quantum biologist.
Exactly. Yeah.
Exactly.
Yeah.
Take care.
Cheers,
Take care,
guys.
Keryn. See you soon. Bye-bye. Thank you.
Okay,
Thank you very much.